And you might be like, okay, if we take glycolysis and that takes glucose and makes energy out of it, well, why do we want to build a new glucose molecule just to build it down, break it down later? Because not only that, but gluconeogenesis actually takes up some of your cell's energy. So it's like, why do we want to invest energy just to break it down and harvest it later? Well, then why would we do that? The thing is that your cells prefer glucose. And remember that little chart I had in a previous lecture where I said your body prefers carbs first, then lipids, then proteins, and nucleic acids and alcohol. So glucose is what your cells love. They love glucose because it's easy for them to use and break down into energy. Now, the thing is that glucose is easily converted to ATP, and the thing is that your brain, special tissues and cells in your body, they cannot metabolize fatty acids or amino acids directly. So those other macromolecules that give energy, they either interfere with their normal processes if they try to metabolize it, or they lack the capability to metabolize that. So I like to think of it like this. Back to my cash analogy with energy in the cell. I like to think of it as like sometimes you encounter a restaurant or business or somewhere that only accepts cash. And remember, like, okay, so I think of like glucose kind of like cash in terms of like nutrients. So the thing is that the red blood cells, they don't like fatty acids. They like glucose, but they don't really like fatty acids or amino acids. Whereas your brain, it can use ketone bodies, it can use lipids, but it vastly prefers glucose of the other ones. Just like how cash is pretty easily used, at least here in the US. I think other countries are moving away from cash, but at least here in Hawaii, like pretty much everyone will accept whatever cash you give them. But same with your red blood cells and your brain. They're a little more picky. They prefer glucose over any other energy source. Okay, so then why do red blood cells prefer glucose? Now go back all to I think it's a blood lecture when we talked about how you make a red blood cell. Remember that the red blood cells, when they become a fully developed erythrocyte, they eject their nucleus. But the other interesting thing is that they not only lose their nucleus, they also lose their mitochondria as well. So red blood cells lack mitochondria. Now, this is very interesting. Why? Because can these processes still occur? Can glycolysis still occur if you don't have mitochondria in the cell? Well, you might have to, it was a very small detail, but very important detail. And this is why it's important to know where glycolysis occurs in the cell. It does not occur in the mitochondria, it occurs in the cytosol. Do red blood cells have cytosol? They still have cytosol, so we still can have glycolysis. Now, the citric acid cycle. And let's ask the chat, if you don't have mitochondria, can you do the Krebs cycle? Can you do the citric acid cycle? Yep, so where does the Krebs cycle occur? It occurs in the mitochondria. So if you don't have mitochondria, you don't have the citric acid cycle. And the citric acid cycle, so no citric acid cycle in a red blood cell. So the thing is that you can still get energy from glycolysis, but red blood cells also use another type of way to get energy as well. So they also use lactic acid fermentation. And quick question, since we get our energy from food, do our cells prefer glucose from sugary foods or from fruits? Well, if you're talking about the same sugar, if you're talking about like say fructose, whether it's from a processed food or whether it's from if it's the same chemical, regardless if it's from a natural or synthetic, your body doesn't discriminate. It's like, okay, is it going to discriminate between glucose made in a lab versus glucose from a plant? It's still going to use the same enzyme. It's still the same chemical formula. It's still going to have the same metabolite, same energy yield. But where it comes interesting is like, okay, what about breaking down things like how much glucose is released when you digest a certain food? Like, or the way these it's like, okay, once it's in that molecule form, not too much difference, but like remember our whole thing with glycemic index and how much glucose you release? That's where it starts to affect like, okay, the rate of whether cells break it down or they try to convert it to other type of nutrients or try to store it as fatty acids. So it's kind of interesting. But yeah, and if you're talking about the same molecule and chemical, it doesn't matter the source. Their cells they can't really tell the difference. All right, so what is this lactic acid fermentation? Well, this is from your OpenStax book, and again, it's free for all. And what we have here, let's zoom in, is there's a lot going on in this. So let's go to the upper part of that. So again, we start with one glucose molecule, and then you break it apart into two 3-carbon molecules, and that's our special pyruvate. And whether there's oxygen present or not. Not only oxygen, but also mitochondria. So if there's yes, we can do everything we talked about in the previous lectures, and if no, then they have to do anaerobic respiration. In other words, getting energy without the presence of oxygen. So if you have oxygen, that's what we covered last lecture. So again, you have to convert pyruvate to acetyl-CoA. Acetyl-CoA feeds into the citric acid cycle in the mitochondria. And then you have the electron transport chain, and the process is called oxidative phosphorylation. And again, remember, this is moving the protons and using the energy from those stored up protons to drive ATP synthase and synthesize new ATP. And the yield, again, for me personally, it's funny because like the OpenStax has 36 ATP, so they use that number. For me, for this class, just know it's 30 something, and I'll get to why in just a little in a little bit. Okay, so what if you don't have oxygen? Or what if you don't have mitochondria, like the red blood cells? Well, now you're forced to do this anaerobic respiration. So what happens there is that pyruvate is converted into lactate instead. And then you also regenerate this NAD+. So what happens is that you basically only get 2 ATP per glucose. What's the take-home point here? Well, how much does that compare to when you had oxygen in the equation? So this is lactic acid fermentation, and it's not the same. You might be thinking fermentation like as in baking bread or making wine or beer. But that's a different type of fermentation. Instead, you produce ethanol in that. So yeast produce the ethanol, aka drinking alcohol. So we don't do that. That's why we don't get drunk when we work out, because we produce lactate. We don't produce ethanol. All right, so what's the big picture here? Well, in both the aerobic respiration and lactic acid fermentation, you start out with the same material. You still have glucose that's converted into pyruvate. So same starting materials, but the difference is that is there oxygen or is there mitochondria? You may have oxygen, but if there's no mitochondria, you can't do the Krebs cycle and you can't complete this full process of aerobic respiration. So with aerobic respiration, you get the carbon dioxide and water, whereas lactic acid fermentation, you produce lactic acid. And again, what's the big difference? Well, you get energy, and again, for this class it's 30 something. I think the older sources are more like 36, 38. Newer sources argue more for 30 to 32 ATP. And with lactic acid fermentation, look at that. Which is greater, 30 or 30 something or 2? Yeah, so this is the big thing. The presence of oxygen vastly improves. Again, you still have one glucose molecule feeding into both equations, but the presence of oxygen and the process of aerobic respiration, you get much more payoff and yield of ATP per glucose molecule compared to lactic acid fermentation. So this is why your red blood cells can still produce energy, but it's a lot less efficient than aerobic respiration.